in record. Ladies, so last time, I can't remember how many videos ago, I did a video of an X-Inactivation, of X-Inactivation, how they prevent us from having parthenogenesis, or just one of the steps anyway. And I have something else for you here, right? So we did this, we talked about this, right? You know, it goes from here, X-Inactivation, which is trying to equalize the amount of XX, right? That the males and females have. And this is because even though they don't try to equalize, they don't, even though like females have the natural XX and males do not, yeah. And the reason is because the X is the actual human essence. So if you have two X's and he has one X, if you have two X's and one X, it means that you are going to be far more human than him. It means you're going to live longer. It likely means, right? It likely means that you are going to live longer. You are going to be stronger. You are going to be what? Smarter, and all kinds of things. And that's basically what that means, right? That's why they needed, that's why I believe that they needed to quote unquote equalize, because when you look at what they call equalizing, he has two active chromosomes, two active sex chromosomes, quote unquote sex chromosomes, but you only have one active one. So he actually is more than you. I believe this is part of why they are stronger. I believe that this is part of why they are stronger. They are stronger and taller, right? But that is just a theory. I haven't actually done any kind of research on that to say definitively so. But I think it has something to do with that. But and that X-Inactivation, it does this, right? So that means that this one X, in the red, it does this. Let's see. So you go from having two active X's to only one active X. And one active X, when I say active versus inactive, in order for you to maintain your cells, duplicate your cells, do cellular activity, you need your genes on a regular basis. They are constantly working. The genes are literally on work 24/7, right? Renewing cells, doing this and doing that. They're constantly making red blood cells. They're constantly making white blood cells. They're constantly making, the proteins that carry out your glucogenesis, which is when you make glucose. The ones that store away your glucose. They're constantly doing that. Your genes are literally constantly doing that. They are the actual machine that you're living on. They are the machines that you are living on, right? Which means that you need your genes. There's no question of, you need only half genes. You need all of your genes all the time. And this is just a falsehood that they're telling people, they're telling students of the universities, because this is a woman here who was saying this, that you need to equalize it, and it's not fair to the boys, and some kind of nonsense like that, right? And so the things that you need, you're not going to not be able to access them, and then think that you will have long health and happiness, long life, health, and happiness. That's just not going to occur. And that, in fact, does explain why we are riddled with diseases, right? Even the healthy ones, there's problems and upon problems on problems, right? So something else came across me today. Again, I'm constantly doing this, doing research like this, and I got something else that has my brain going haywire. Okay. It is this one. I got this one. I woke up to my FIA.
When you look at a star,
page,
you're looking back in time. But I see the future.
talking about this. Talking about
this
this.
lecture, I'd like to think about the stage.
This is just a deeper dive into the same very topic, right? So it's talking about how it is talking specifically about the genes. Now, it's talking about the genes that makes this process happen, the X inactivation, and oh, by the way, actually, let me finish what I was saying about the activation versus the inactivation. So the activation versus the inactive X, the active X versus the inactive X. So the active X is the one that it is a machine that you're living on. You are constantly, running programs off of that. You're constantly running programs off of that, right? And the X and the second one, that one is not available for you to run programs. That's what that means, right? So even though you're using your genes every second of every day, right? That means that you need them to be available for you to use. The second X is not available for you to use. It is literally like on lockdown imprisonment. This is how this is part of this is the first step on how they blocked our ability to parthenogize, right? So now here is a deeper dive on how that happened. And I just want you to
stages of X chromosome activation. So we know that X activation doesn't happen instantly, but instead happens progressively over a period of developmental time. And during this time, we know that there are specific stages that X activation needs to go through. So the first stage of X activation is counting. So what I mentioned earlier was that in karyotypically abnormal cells, we know that there's only going to be one X chromosome that remains active per diploid set of autosomes. And so the cell must have some mechanism to count how many X chromosomes there are in relation to how many autosomes there are within the cell.
Okay, so the autosomes is the sex chromosomes versus the non-sex chromosomes, right? So the autosomes are the non-sex chromosomes, right? And the XY are the sex chromosomes, right? And the XY, they are just they're just the last pair, or rather the first pair. I mean, they're counting them backwards, but the X is actually the first. The X is actually the first because it is the one that determines your humanity. It is the one that has the blueprint of your life. It is the one that has your actual life. Without the X, there wouldn't even be a cell to make all of you, to make literally all of you, because all of you are cells, right? So without the X, the cell dies. Without the X chromosome, the cell dies. Okay, so there won't even be life left for the cell, right? So which just tells you that the cell itself is just a bunch of things, and it is held together by the X. So when they're saying that it is, like it's, so we have 23 pairs of 46 chromosomes, and the sex chromosomes are just a pair of that, right? So they're basically counting. So this picture that she's talking about is the number of those sex chromosomes versus the number of the autosomes, right? So where we have two sex chromosomes, we would have 44 autosomes. Does that make sense? To make a total of 46 chromosomes. To make a total of 46 chromosomes. And those 46 chromosomes are divided into 23 pairs.
And this is the mechanism of counting. The second stage of X-inactivation is choice. So I mentioned imprinted X-inactivation in the last lecture. And clearly here, there is no choice. The choice has already been predetermined that the paternal X chromosome will be silenced. However, in this instance, choice means that in random X-inactivation, there must be a choice about which of the two X chromosomes in a normal female cell will be the one that's selected to be inactivated. The third stage of X-inactivation is the initiation. And really, this is all about the expression of that long non-coding RNA that I mentioned in previous lectures called exist. Exist expression comes from its particular location on the X chromosome known as the X-inactivation center, which we'll talk about in future lectures. After this initiation of exist expression, then the exist needs to spread to cover the entire X chromosome or become inactive. And following on from that, we need to have establishment of the silencing marks. And finally, after that, we'll have the maintenance of these inactivation.
So she's saying that long coding RNA. Again, our human DNA is in DNA. It is not in RNA. And DNA is different from RNA insofar as the molecules that make up one of the foundation of the molecule, which is the molecule being DNA or RNA, right? So DNA has a different kind of molecule and RNA has a different kind of molecule as its foundation, right? So I don't understand why that didn't trigger them to research where an RNA like this would come from, like to test its originality to the human species, right? Because there was something else that I noticed as I was doing this research for the first video. Okay, I noticed that the 23 pairs of chromosomes that I just talked about, right? Every single pair is identical in size and shape. Every single pair in the human DNA, in the human genome, every single pair have the same size, the same shape, which is, let's say for instance, 1 versus 15. 1 is kind of long, right? The pairs of 1 are both long and lanky, and 15 is kind of like short and stocky, right? So they have different shapes, but each pair is identical. Each pair, both chromosomes in each pair are going to be identical, except for the sex chromosomes. But it's not just the sex chromosome, it's the sex chromosome specifically in the males. Specifically in the males. The X in women. Our chromosomes are all identical. The XX, every single pair of chromosomes are identical. Is identical. But in the males, only the sex chromosomes, their sex chromosomes are not identical. Do you guys get this? Are you guys getting this? This is just yet another way I've been able to identify that they are not human. They are not human. The reason the chromosomes, the chromosomal pairs are identical, right? It's because so that they can read each other. There is a thing, when you're duplicating the chromosomes, there's a thing that controls them, right? Which is it's called a telomere cap. God, this is so much science, which I was only able to understand because I had a science background already, which even though I hadn't done it in decades already, right? I already had a science background, doing a deep dive into biology and biological and scientific publications. So I was able to understand a lot of this stuff, right? So there's a thing when you have your chromosomes. The reason they are the same size is because there's a thing on them that controls, how do you know when to stop? How do you know when to stop duplicating, right? Yeah. So they have a telomere cap, and so they're kind of reading each other, right? So the telomere cap, the way it duplicates is that in order for it to, because DNA is continuous, so it has to cut off the telomere cap, which means it shortens the telomere cap, but you need the telomere cap in order for your lifespan to be maintained, right? So my guess is, I haven't looked into this. My guess is they are the same size so that they can cap each other off. If you cut one off in order to maintain it, in order to know where to stop, right? In order to know where to stop, you have to cut off a little bit of the DNA of the telomere. And when the telomere gets short, if you continue to cut it, but you don't put it back in somehow, right? The telomere will eventually end. And when the telomere ends, the cell will die, right? The cell will die. So, which means that if that is happening all over your body, you will eventually die, right? So I believe that the reason they're the same size is that they are capping off each other. When you cut the telomere, one will repair the other one. They will repair each other. They will do like a self-repairing thing, right? So that the telomere is maintained, and that is how it keeps the cells from dying off. I believe that. I still have to look into that, but that is just like a wild theory that I'm thinking out there, right? But I know that the reason that they're the same size, it has a very, very significant reason. Now the males don't have that. The males don't have that, which means they're sex chromosomes, right? Which means that when they are, which explains why they kind of have a shorter lifespan. This is why they have a shorter lifespan, right? You aren't supposed to have that. If you have 46, if you have 46 chromosomes in one of you that are all identical in size and shape, it's because they need to be. Biology does not make a mistake when it designs things like this, right? It's because they need to be, which means that the Y not being the same size as the X, that is very significant. That is very significant. That us having one X inactivated, that is very significant. I believe that is why our lifespan is so much shorter than what you read in the Bible of Abraham, of Sarah, of what was this guy's name? The guy of the ark, of Adam, of Eve, etc.
And maintenance is, of course, perhaps the longest lived phase of X-inactivation, because while the first few stages happen just within a few days in the embryo, maintenance must occur for the life of that female organism, and so can be occurring for many decades, up to perhaps 100 years. So while
so you need to understand what she just said, because this is very, very significant. The fix that they did to us, it is so not innate to us that they have to keep doing it in order to keep it working. It is so far from what is normal that they have to keep doing it in order for it to be maintained. This is yet another sign that we can get out of it. Because once you disrupt this maintenance structure, it would eventually go back to what it's supposed to be. That's why it has to be maintained. This is how they keep us locked down.
While we'll go through many of the molecular events that are involved in each of these stages, I think the really key thing to understand and remember is that the way X-inactivation happens is that there's a progressive layering of an accumulation of epigenetic marks. And it's by this progressive layering of redundant epigenetic marks that you can ensure this mitotic heritability and the incredible...
Yes, that is exactly what I've noticed before I even found this video, that it had to be layered because it was so unnatural, because because of the complexity of the control systems to make sure that this organism that is being produced by these genes are going to work every time. It's going to work the right way every time, right? So they have a lot of control system, which means that if they're getting an X, a cell that is X inactivated, it will kill it. It will kill the cell, and you can never make a child after that. Do you understand? So the system of control to make sure that the organism that you are making comes out right every time, right? It's so complex that, one of them would be that if it sees an X, a cell, if it sees all the cells with only one X activated, it would kill them. It would kill the offspring, which means, and then if it's happening to every child, right? It means you could never get a child, right? So they have to layer it to make sure that it doesn't kill this, right? It doesn't kill the child, over there, and it doesn't kill so that it doesn't, so that the thing basically works. Right. And that is just, and the control, the quality control system, that is just one way that makes it, that is just one way that, that makes it work. The other way is that, the other way is that even when it would, in some scenario, it would kill the cell, but in other scenarios, it would simply fix the cell. In some scenarios, it would simply fix it. It would simply send out, fixers to, fix this and fix that and fix this, right? So that's another way to make sure that things work correctly. In some ways, it would just kill the entire thing, and in some other ways, it would fix it, right? So you have to make sure you have to have control systems to override the fixers as well, right? So you have to have a control system to override the killers so that you can actually get a child, and then you have to have an override system for the fixers so that it doesn't fix what was wrong, what they are trying to break, right? Exactly.
Incredible stability of X-inactivation, and that's why it can last for the lifetime of an organism. So before going into all the details for each of these different stages of X-inactivation, the most important thing to remember is that it's really all about exist. So exist is the critical determinant for X-inactivation to occur. This long non-coding RNA is essential for X-inactivation. And so the first few stages of X-inactivation, counting and choice, are really about getting exist expressed in the first place. And the latter stages of X-inactivation are turning this expression of exist into transcriptional silence. And so just keep this in mind at all stages. So before we go any further, let's have a little refresher about what exist is. So as I said, exist is the critical determinant for X-inactivation. Without exist, nothing else can happen. So it's a 17 kilobase long non-coding RNA. It's spliced, just like a normal RNA would be. It's polyadenylated, but it's constrained to the nucleus. It isn't exported from the nucleus to be made into protein. And indeed, there's no open reading frame to be found. So there's no protein product that's produced. We know that exist is expressed from just one of the two X chromosomes in a normal female cell. And it coats that X chromosome in cis. That is, it coats the X chromosome from which it is expressed, but doesn't act on the other X chromosome. So the first detectable event in X-inactivation is indeed the expression of this long non-coding RNA. So here shown on the right is an image that I've shown you before, showing the DNA first of all in the nucleus, which is stained in blue. And then we also have shown in yellow the exist DNA itself. Of course, there are two pieces of exist DNA, one on each of the two X chromosomes. So in addition to these two exist DNA foci, which you can see with the very small yellow dots that are shown indicated with each of these arrows, you can also see the exist RNA, which is stained in red using in situ hybridization. But this exist RNA is not found on both X chromosomes, but instead just found on the inactive X chromosome, where it forms a cloud. So it's not a punctate RNA like you would expect if you just had a small amount of RNA being expressed and then being exported, but rather it's being expressed and retained in the region from which it's expressed. And so it forms this cloud which actually coats the entire X chromosome, which will become inactive. When it's Christmas time in Texas. So let's now think about X chromosome counting, this first stage of X-inactivation. Well, there still isn't a lot known about X chromosome counting, and over the years there have been many theories about how this might occur, many of which have been controversial. But I think now the consensus is that there are both X-linked factors and autosomal factors, and it's the balance between these X-linked factors and the autosomal factors which allows the cell to be able to count how many X chromosomes there are compared with how many autosomes.
In other words, even though the reason you have the chromosomes that are the same size, the same the same size, the same shape, right? It's because you actually need it that way, because that is such a very specific thing. That is because that is done because you need it that way. It needs you to not have them like that in order to work. So it has to disrupt the number of that structure as insofar as your sex chromosomes are concerned. This is really diabolical. This is the diabolical design.
So far we know essentially nothing about these autosomal factors, but we do know something about the X-linked factors. It appears that there are many regions that are quite close to the exist gene itself that encode non-coding RNAs. These particular DNA elements, which I'll show you in the next slide, are all regulating exist expression. And we know if we do something to manipulate these different elements, we get the incorrect number of X chromosomes being inactivated. And so they must have something to do with counting. So let's look at those factors now, these DNA elements. So what's pictured here is the X-inactivation center. It's around about 100 kilobases in size, although this picture isn't perfectly to scale. And at its center is the exist long non-coding RNA gene, here shown in blue. In the darker blue,
so the center is the entire area of that causes this behavior to happen, right? and as opposed to the gene that actually does it, right? So the other ones are basically like the control systems. They're controlling this and they're controlling that, and they're preventing this and preventing that, but it is the actual gene that does it.
Blue small box, I'm indicating the repeat A, rep A, and this is the region. This is a region of exist, which is very important for silencing because it binds to polycomb repressive complex 2, as I've mentioned before, and I will come back to again. But all of these other elements that are shown here, all of these other genes, also contribute to the expression of exist. They all control exist expression in some way, shape, or form. So in general, the ones that are shown in the yellow to red tones that are down this end of the X-Inactivation center are involved in repressing exist expression. Whereas the ones shown here in green, FTX and JPX, are involved in activating exist expression. Now, what we know is that this X-Inactivation center, if you take this region in its entirety, is necessary and sufficient for X-Inactivation to occur. So what I mean by that is if you delete this region, then X-Inactivation won't occur, therefore it's necessary. However, if you take this X-Inactivation center, this whole region, and place it onto an autosome, then you will get inactivation of at least part of that autosome. And so it's sufficient to get this inactivation to take place. We know that we need more than just the exist gene itself. Instead, we need this whole region with all of these other elements. This can also be seen because in some patients, there are translocations between the X-Inactivation center and autosome. So it's not just through experimental methods that we've placed the X-Inactivation center on another autosome. So what are all of these different factors doing and how are they controlling exist expression of elements? We know that we need more than just the exist gene itself. Instead, we need this whole region with all of these other elements. This can also be seen because in some patients, there are translocations between the X-Inactivation center and autosome. So it's not just through experimental
Translocation.
We've placed the X-Inactivation center.
Translocation, this is very important. Translocation, this is also very, very important. This is some really deep, godly stuff right here, okay? Translocation is when genes move from one place to another. Okay, so where you can see this is right are females that you see visually are females, but they act like they are male. That is a gene translocation from the Y to the X. And you can see the same thing on males, right? You can see the same thing on males, where something will have happened to cause, genes translocated from females to male. Translocation, this is what the presence of the Y chromosome has caused such havoc on our gene pool. We don't even know. We don't even know. This is why when they tell you vaccines do this and vaccines do that, BS. BS. Now when I know this is going on, that is some BS. Now when I know this is going on. Gene, now here's the thing that you don't know. Okay, I hope, I think I actually said it in one of the previous videos that I did of talking about this very X inactivation issue. The X chromosome has a way to fix itself because it made its survival based on self investment, which is it invests itself in being better, being all the shit, right? Being all that and in a bag of chips, right? The Y chromosome doesn't invest in itself because it made its survival based on being able to use other organisms to do its job. So it doesn't have to perfect its own genes, right? So what happens with the Y chromosome is it mutates a lot. It mutates a lot. And the reason the X does not is because it has a mechanism to fix itself. It actually evolved a mechanism to fix itself. So now imagine if you have the X chromosome and you have two of it that are fixing each other and fixing everything else. Do you think it's possible that you might not actually have any diseases, that you might not actually, die at 100 years old, die at 80 years old, die at 50? Do you think get all this autoimmune diseases? Do you think that's possible? Do you think that you might prevent cancer if you have two X chromosomes that are fixing everything? Hmm? Do you think that? Do you think those two chromosomes, do you think if they're smart enough to evolve a way to self-heal themselves, right? To self-heal themselves, do you think that they can communicate to heal each other? Ooh, 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 ooh.
Center on another autosome. This can also be seen because in some patients there are translocations between the X-Inactivation center and autosomes. So it's not just through experimental methods that we've.
So that means that if a translocation occurred, if a unsanctioned translocation would have occurred with two active X's, that would have been fixed. But with the Y chromosome, it doesn't. But with the Y chromosome, but without an X, right? Without a second X, that problem couldn't be fixed. Yeah. And with the Y chromosome as your second chromosome, that can be fixed. And with your second chromosome, if you're a female, if your second chromosome is inactive, do you think that could be fixed? No, it can't.
Place the X-Inactivation center on another autosome. So what are all of these different factors doing and how are they controlling exist expression? Well, in all cases, they are long non-coding RNAs. So this center is made up of a bunch of non-coding RNAs which control another non-coding RNA. I'm not going to go through the specialized features of each one of these, but perhaps the most important one to remember is TSIX or T6. This long non-coding RNA is transcribed antisense to exist, and it represses exist expression. Whereas DXPAS34 and X-site themselves activate T6, whereas DXPAS34 sense TSIX or T6. This long non-coding RNA is transcribed antisense to exist, and it represses exist expression. Whereas DXPAS34 and X-site themselves activate T6 and thereby indirectly repress exist expression, because they do so by activating T6. So again, these precise details aren't important, except to remember that we have a predominance of factors which repress exist expression on code B. Or R-lim, it has two names, RNA, another gene called R. If we look upstream of this X-Inactivation center, a couple of hundred kb upstream, we find another gene called RNF12. And this gene RNF12, rather than being a non-coding RNA, actually does code for a protein. The protein is called RNF12 or R-lim. It has two names that are interchangeably used. So this gene RNF12, being that it's on an X chromosome, is subject to X-inactivation. What's very interesting was that in 2009, a real breakthrough.
This is what I actually want you to hear.
In our understanding of X chromosome counting. And this was a discovery for this gene, of a role for this gene RNF12, which sits a few hundred kilobases outside of the X-inactivation center. So what was proposed and shown was that you needed a threshold level of RNF12 for X-inactivation to begin to occur. So I'm going to explain this in a series of diagrams. So to start out with, if you consider that you have cells with two active X chromosomes, then the female nucleus has two copies of RNF12 and the male has one. And therefore, with two active X chromosomes, you'll have twice the number of molecules of RNF12 here shown as a red blob. So this is RNF12. You'll have twice the number of molecules in a female nucleus that has two active X chromosomes compared with a male nucleus which has only one.
You have to understand that everything that your biology is doing, because the X has a way to fix itself, anything that it's doing is because you need it, right? So let's continue.
However, then once X-inactivation proceeds, you will inactivate one of those copies of RNF12 and then have only the same amount of RNF12 as in a male cell.
Now you have half of the proteins that you need.
And so this is why it was proposed that there must be a threshold. So if you had what was shown here as 8 molecules of RNF12 protein, that would be above the threshold and X-inactivation would be initiated. Whereas once you have 4 copies as shown now, this would be insufficient to initiate X-inactivation. However, although this was really a groundbreaking discovery, it still isn't enough to explain the mechanism of X chromosome counting completely. And that's for 2 reasons. First of all, we know if you completely delete RNF12 in a female cell, X-inactivation will eventually occur. It will occur much later than it should have done, and it occurs very inefficiently, but it can occur in some instances. And so this suggests that there are indeed other factors that are still important in counting, and that these can in some way compensate for a loss of RNF12. So the question is then, how does RNF12 actually work? So how is it really going to bring about this initiation of X-inactivation? So what we think is that RNF12 activates exist. So just like those other two green elements, those long non-coding RNAs, RNF12 also activates exist expression, but this time as a protein activator of exist expression. We think that male cells have insufficient RNF12 to activate exist, and after X-inactivation proceeds within a female cell, we believe that we've reduced the level of RNF12 to such a level that the second X chromosome will not be inactivated because we are below the threshold level of RNF12. So clearly, RNF12 will somehow be involved in this counting process, this first stage of X-inactivation, but we still don't know, and this is by the levels of its own expression, this threshold, but we really still don't know anything about the autosomal factors involved, and we don't know anything, and it's clear from the RNF12 results that there are other factors that are also involved in counting, but they're yet to be identified. In the next lecture, we'll think about X-inactivation and pluripotency, which I've mentioned in.
Okay, so that's kind of the end. So did you get the part where she says that RNF12 also gets inactivated, right? Okay, so now I told you that biology does not make, no, not that it doesn't make mistakes, right? It does, right? But that when it designs something, the design that it doesn't make a mistake in that. Okay, and I just told you that because the X's, they have a way to fix themselves, right? Which means that they've lived for millions of years. That means that whatever it is that they have now, you need it. You need every single thing that they are doing now, right? So this is what I was thinking when I thought this question. So what happens if you deactivate, if you inactivate one RNF copy, right? And then the other one mutates and it doesn't get corrected. What happens? What happens? Like, how much do you not need it, right? How little importance is it that you would think it would be okay to inactivate it? Let's see. Okay, this is not to say that what does it do, right? This is not to say what does it do, right? There's a difference between what happens, right, and what does it do, right? So I kind of want you to get clear on that. Whew. So let's look at what it does. What happens when you deactivate? What happens when you lose that one copy? Which is what would happen as the consequence of all of these things. Not just as a consequence of deactivating the X, but as a consequence of all of this. And as a consequence of the nature of genes, which is that when you replicate, which is all the time, right? When you replicate it, which is all the time, because you have to renew your cells all the time, yeah? Mistakes happen. Replication is not perfect. That is why the X evolved a way to fix itself, right? So that it fixes those mistakes so that it can make sure to have a perfect organism every time. Okay, so what happens? Let's see. Okay, so I looked up, I did this search. I can't remember what search I did, right? I can't remember what search I did, but I got this here, publication, that talks about what happens when RNF12 has a mutation. So let's read what it says. It creates a Tokus disease, right? Tone-Caucher syndrome. I don't know if I'm saying that right, but we're going to go on. It's an intellectual disability. Intellectual disability. Now, when the men tell us that we are dumb, they are they are better strategists, they are this and that. Now, if you have double the amount of something whose absence causes intellectual disability, do you think you might actually be smarter than the males? Not just on like intellectually, not just intellectual, how should I say this, smart, but strategically smart, right? Do you think you might? Yeah, I think you might. So, tokens is caused exclusively by variants in the gene encoding the E3 ubiquitin ligase, blah, 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 also known as RNF. What's telling you that it is this gene, this RNF gene, and it's telling you, kind of what it does, right? Yes, also known as. So, tokens is caused exclusively by the variants in the gene known as RNF12. That's how you read that. Here we report the identification of a novel RLM missense variant. That means it's new, right? It's new, not typical, I think. And missense, like it missed something. Adjacent to the regulatory basic region, which causes a severe form of TOCUS, resulting in perinatal lethality by diaphragmatic hernia. Ooh, perinatal lethality. It's telling you, I actually don't know what perinatal means, so let me look up that word. I'm guessing it says before, like in the zygote, either in the zygote or embryo, before it becomes a baby, I think. Perinatal. Let's see. Relating to the time, usually weeks, immediately before and after birth. Oh, okay. Perinatal. So I was wrong. So let's go back and read that. So before and after birth, lethality, resulting in perinatal lethality by diaphragmatic hernia. The diaphragm, right? It causes death by a hernia in the diaphragm. Do you see that? It's lethal. It can cause lethality. It's not that it's lethal. It can cause lethality. Yeah. So you have to understand that the absence of it is telling you how important that thing is, which is why you would need 2 copies of it. I know this biology like it's the back of my mind because I studied how it works, not just in things that we know, but in things that we don't know. That I'm like, how would this work if this were to actually work? And it was the same every time. It was telling me something every time. The reason you have 2 copies of something is because you need 2 copies of it. If it's going to be fatal, if it's going to be lethal, having 2 copies of it is a backup. Is a backup. This is what they're taking away. Inheritance and X-chromosome inactivation patterns implicate RLIM as the likely pathogenic variant in the affected individual and within the kindred. We show that the RLIM variant disrupts both expression and function of the protein in an embryonic stem cell model. That's right, it starts from when it's an embryo. RLIM is correctly localized to the nucleus, but is readily degraded by proteasome. RLIM variant also displays significantly impaired E3 ubiquitin ligase activity, which means the major part of what this protein RNF does. This mutation disrupts it. which interferes with RLIM function in cyst long non-coding RNA induction. Okay, so that is saying that. So just like that woman was saying that RNIF, contributes is necessary as part of the initial stages of this X inactivation. That's what it's saying. So when it mutates and it's not even in its right state, it even disrupts this one. It disrupts the X inactivation process, right? Which means the inactivation process is after this RNIF. Where is it? The RNF. Yes. So this what you call it? This inactivation process is after this gene. It leaves its counterpart alone. Which means it's going after the correct thing. It's going after the correct thing. It is targeting something that is correct. Oh my God, do you guys know how impossible biology is? That is. Do you guys understand how impossible this is in biology? It is targeting the correct thing. Oof. Our data uncover a highly disruptive missense variant in RLM that causes a severe form of TOCUS, thereby expanding our understanding of the molecular and phenotypic spectrum of disease severity, which means that it is seriously severe. It has a spectrum, right? And the highest part of the spectrum is actually really, really bad. This is what they are disrupting. This is what I wanted to show you. That the so-called X thing that they're telling you, they're trying to fix it. Actually, let's listen to that part. Listen to that part. It's right here. Okay. Let's listen to that part. Listen to it. Let's listen to the BS that we are being fed by the medical and the science community. Okay.
Chromosome inactivation or X-inactivation, which is also called lionization, after the scientist Mary Lyon, who discovered it in 19. Whatever genes are present on X chromosomes, female have two alleles of each gene, right? Because there are two chromosomes, so each gene has two alleles. But in male, there's only one X chromosome, so that means there's only one allele for each gene present on X chromosome. So that means there is inequality in terms of genes that are present.
Equality, he calls it. Inequality. Kind of like the type where, if you have two eyes and someone has one eye, there's an inequality of eyes. So you should blind the person with two eyes. That kind of inequality.
On X chromosome for male and female. So to balance this out, for dosage compensation.
Balance it out. So this is what they're teaching them. This is what the men, you have to understand, it is the men who are controlling these things. They are the ones who established it. And now women are just following it because they don't understand that something is deeply wrong, that it is anti-female, that it is female hating, right? They don't understand that. They are literally saying their understanding, the men's understanding is that if the women are better than us in any way, we should hobble them. That is what is fair. That is what is fair. We should impair them. That is what is fair. That is what is fair. Right? They're trying to do that right now with reproduction. I have read in 2 days, information about, how men need reproductive equality. How men need reproductive equality, even though they don't carry children. They want to be equal to you. They want to have the same rights. They want to, and that can only be achieved by stopping you from aborting, which is going to kill you, right? Right? And then getting the right to not give you money for any child that you do bear. That is what they call equality. That is what they call equality. It's not equality based on what you can actually do. It's equality based on hobble the other person so that I can get ahead. These things are demonic. You ain't gonna tell me otherwise. This shit is fucking demonic.
To have a balance in expression in terms of genes that are present and for
Have a balance of expression, because God forbid you have just 5 more heirs than them. They want to bald you so that they can be, that's what they call equal.
expression in both male and female. There is something called X-inactivation occurs in case of female. That means one of the X chromosomes in terms of genes that are present and for expression in both male and female. There is something called X-inactivation occurs in case of female. That means one of the X chromosome would be inactivated. All right. Now, that is one thing, that there is imbalance because there are 2 X chromosome in female and there is 1 in male. Also, there is no need of 2 X chromosome. There will be double expression of the same.
There's no need for 2 X chromosomes. Well, you don't need a full head of hair. If you're gonna be bald, you should be bald too. If you're gonna be bald, you should be bald too. If I have only 1 leg, you should not get any because, well, you don't need them.
Genes, double proteins, double products will be formed, which are not needed by these cells. So only 1 X chromosome.
That's what they're telling you you don't need. That is what they're doing. That is what they're telling you they are doing. They are doing this thing that causes that problem. Intellectual disability, right? Lethality, that could kill you. They're causing that problem that could kill you in order to cause what? An equality. An equality in, with them. Which means that, and you can see how they are bigger and stronger than us, right? How they are bigger and stronger than us. How come they didn't cause equality there? How come they didn't cause equality there? This is demonic behavior.
Chromosome has to or needs to be expressed. So this is what we are going to see now, how this X inactivation occurs, how this balance takes place. So let's begin. Now what happens in the zygote, when it's, if it's a female zygote, that is XX chromosome, in the zygote there will be 2 X chromosome present. Okay. Now as the zygote divides, during the embryonic development, what happens is in each of the cell.
What did you hear? What did that one say? It starts in the embryo, because that is when this demonic thing happens, right? So before this, they won't find that it happened. They won't. Where's this article? Where is this article? Okay, so they won't find this before the embryo stage, because then you have all your X's, which actually explains to me why why babies terminate, why you would have unviable babies at all, right? Because unviable babies, the reason they eventually terminate is because they're not viable. And typically, they're not viable because once these things happen, right? Because before this happens, everything works, right? Everything works, and then this happens, and so things start to break because they broke it. Yeah, this is why. Otherwise, listen. What would happen is typically when you have a, an organism that is complex, the reason our body can, can sustain our cells, our eyes, our hair, our heart, our lungs, our brains, our, muscles, and all these things, all these cells, right? Trillions and trillions and trillions and trillions and trillions and trillions of them for an extended period of time is because they invest in literally perfecting their quality control system. Do you understand that? Because there's too many things going on for you to not die so quickly when, if that is not what happens, right? There's too many things going on that you would just be getting sick, left, right, center, up, down, right? They invested in perfection. Their game is perfection, right? And you might not you would not know that, because since they broke the system, it started to allow a lot of controls, right, to fall by the wayside, right? A lot of quality control steps have to fall by the wayside, because in order for a 1X individual, which is a male, to even exist, in order for a male to exist, right? In order for a male to exist, the ability to only select for X axis had to have been destroyed. Let me repeat that, okay? In order for the male to happen, because one of the controls, if your organism, if human is female and female is human, that means that you do not need to reproduce any other version. You don't need an XO individual. You don't need a triple X individual. You only need 2X individual, which means that means that you would check for your quality control path would be only XX. Only 2X's would pass quality control. Everything else would just, would either be fixed somehow, or it would be, a third X will be dissolved, or a second X will be like one with an XO cell, an XO embryo, no, excuse me, an XO zygote, right? Would probably have it fixed. Would probably have the X either fixed or the cell destroyed entirely, right? And the same thing would be a triple X. The third X would either be dissolved, right, or it would be or the cell, the zygote would be destroyed entirely, right? Which means that in order for an XY male, which is a singular X male, to go through the quality control system, it has to destroy that system that only approves double X's or only approves 2X's. You have to understand that, right? It has to destroy that control system that only approves 2X's. That is the only way you can produce a male. If there is a quality control system, which there will be, in order for you to be able to produce an organism as complex as us, right? You have to have a perfect quality control system, which means it would in fact have a check for XX. It would have a check for XX. And in case you didn't notice, that counting system that we just talked about, okay, that would be part of that system. That would be part of the system. So it would count for XX, right? And then if it's more than X, it would do something. It would take this action. If it's more than 2, it would take one action. If it's less than 2, it would take another action, right? But if it's if it's 2X's, then it will send it down. It will send it downstream to the next step of development. Yeah. So in order for the XY male with a singular X to even pass through It had to destroy that system. This is why we even get the XO individual, which is the Turner syndrome female. Whew. And this is also why we have triple X females. This is why we have XY females. This is why we have all this, LGBT stuff. This is what is causing it. This is what is causing it. Whew, Jesus Christ. Anyway. Yeah. So I just wanted you to know that. I just wanted you to understand that the fix that they are trying to equalize here, they're not equalizing anything. They are hobbling women. They are hobbling women to allow them to prey on us. They are disabling us to allow them to prey on us. And that disability has far-reaching consequences. It is what is destroying our gene pool. It is what is causing people to be disabled, people to be ill, people to die of cancer, people to do all these things. All these diseases that you're saying, that is what is causing it. And this is just one. This is just one of the steps. This is just one of the steps that they did. That woman was talking about how they layered it. Yeah, how it was a layered approach, and we haven't looked at the other layers. Even in this just one thing, right? It was layers, but there are other downstream quality, control systems to enable this demonic thing, right? And we haven't even looked at that. Look at what is already happening. Look at what is already possible. Lethality. This thing that they're telling you they're turning off, right? You having one copy that can go that can mutate causes lethality. That means it is impossible for what they're fixing to actually be fixed. That's what I'm showing you right here. That is the point of this video. What they're fixing is not a fix. They're not fixing anything. They're destroying it. They are not fixing anything except their ability to prey on us. What they're doing, and in order to do that, they are destroying us. That is the point of this video. What they said they are fixing, they are not fixing. When they said that they evolve, when they said that humans evolve, they mean just them. It had to have taken evolution after evolution to be able to do all of this because it is so complex. They are far simpler than we are. So it has to have taken them, generations after generations after generations after generations to be able to do this. To be able to do this. And I believe that they finally realized that having two X's is making us invincible, right? And that is why they had to what? They had to kill it. Not kill it, inactivate it. And they also realized that it takes two. They also must have realized that at some point that it takes two X's to have a child because they couldn't have a child, right? It takes two X's to have a child, which is why they didn't kill it. Which is why they didn't kill it, because the Turner syndrome females, the one with the just one X, right? They don't even have to inactivate one of their X's. They simply don't have one, right? They are usually barren. They are barren. Anyway, I'm going to end it here for now.